Hello everyone. In the last session, we have discussed about what is pollination, the different types of pollination, the agents responsible for the pollination, the different types of the flowers based upon the pollinating agent, mechanism involved in the pollination with reference to Vallisneria, which is an aquatic plant, and we have also discussed about the pollen-pistil interaction. In today's session, we will discuss about the double fertilization and its further process. So before I begin the session, we will have a small glance on pollen-pistil interaction. Once a pollen tube gets landed on the stigma, it will absorb the water from the stigmatic surface. The pollen tube now germinate and emerges out from the germ pore. The pollen tube penetrates into the stigma, grows downward through the stylar tissue. The pollen tube passes through the style, enters the ovary, and penetrates the ovule in three possible routes. These three possible routes are technically they are named as porogamy, chalazogamy, and mesogamy. So what is a porogamy, what is a chalazogamy, and what is a mesogamy? See, if you look at these three conditions, what we call as the porogamy, or it can be the chalazogamy or mesogamy. If you take the porogamy, porogamy means if the pollen tube enters into the ovule through micropyle region, it is called porogamy. So this porogamy, it is very common and it occurs in more than 80% of the angiosperm. If you look in terms of the mesogamy, if the pollen tube enters the ovule from any region except the chalazal and micropylar region, it is called mesogamy. Then what is chalazogamy? Chalazogamy means if the pollen tube enters into the ovule through the chalazal end, it is called chalazogamy. So what is a porogamy? Porogamy andre ಏನು? If the pollen tube enters into the ovule through the micropyle region, it is called porogamy. So porogamy andre pollen tube micropyle region ಮುಖಾಂತರ ovule ಒಳಗಡೆ entry ಆದರೆ it is called porogamy ಅಂತ, and it is very common in more than 80% of the angiosperm. So mesogamy means what? So mesogamy andre ಏನು? If pollen tube enters into the ovule from any region except chalazal region and the micropylar region, it is called mesogamy. Okay, pollen tube ಅದೇನಾದರೂ ಈ ಚಲಾಜಲ್ ಪಾರ್ಟ್ ಮತ್ತು ಮೈಕ್ರೋಪೈಲಾರ್ ಪಾರ್ಟ್ ಬಿಟ್ಟು ಬೇರೆ ಕಡೆಯಿಂದ ಎಂಟ್ರಿ ಕೊಟ್ಟರೆ ಆ ಓವಿಲ್ ಒಳಗಡೆ ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಮಿಸೋಗ್ಯಾಮಿ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ಚಲಾಜೋಗ್ಯಾಮಿ ಅಂದರೆ ಏನು? ವಾಟ್ ಇಸ್ ಚಲಾಜೋಗ್ಯಾಮಿ? ಇಫ್ ದ ಪೊಲನ್ ಟ್ಯೂಬ್ ಎಂಟರ್ಸ್ ಇನ್ ಟು ದಿ ಓವ್ಯೂಲ್ ಥ್ರೂ ದ ಚಲಾಜಲ್ ಎಂಡ್, ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಚಲಾಜೋಗ್ಯಾಮಿ. ಸೊ ಚಲಾಜೋಗ್ಯಾಮಿ ಅಂದರೆ ಪೊಲನ್ ಟ್ಯೂಬ್ ಚಲಾಜಲ್ ಎಂಡ್ ಮುಖಾಂತರ ಓವಿಲ್ ಒಳಗಡೆ ಎಂಟ್ರಿ ಕೊಟ್ಟರೆ ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ದ ಚಲಾಜೋಗ್ಯಾಮಿ ಅಂತ ಹೇಳ್ತೀವಿ. So now we will discuss about the double fertilization. So what is double fertilization? Double fertilization hingagutte. See, as the pollen tube enters the embryo sac, the pollen tube discharges the two male gametes. One of the male gamete fuses with the central cell that carry two nucleus. Thus, here the three nucleus are involved in the fusion, hence the process is called triple fusion. The triple fusion is always leads to the formation of the primary endosperm nucleus. The other male gamete fuses with the egg cell, resulting in a diploid zygote. This represents the true fertilization, also called the syngamy. Since there is union of the one male gamete with the egg cell and the other male gamete with the central cell, it can be called as double fertilization, and double fertilization is one of the characteristic features of the angiosperms. So what is double fertilization? Double fertilization means what? Double fertilization means the process of a union of one male gamete with the egg cell and the other male gamete with the central cell is called double fertilization. I'll repeat once again. So if you come across a double fertilization, so as the pollen tube enters the embryo sac, the pollen tube discharges the two male gamete. One of the male gamete fuses with the central cell that carry two nucleus. Thus here the three nucleus are involved in the fusion here, and the process is called triple fusion. The triple fusion leads to the formation of the primary endosperm nucleus. On the other hand, the other male gamete fuses with the egg cell resulting in a diploid zygote. This represents the true fertilization, which can also be called as syngamy. Since there is a union of one male gamete with the egg cell and the other male gamete with the central cell, the condition is called double fertilization, and double fertilization is one of the characteristic features of the angiosperms. Let us study the next concept of the development of the endosperm. So, how are endosperm produced? After fertilization, that is when a male gamete fuses with a central cell having a secondary nucleus, it results in the formation of the primary endosperm. This primary endosperm undergoes a series of mitotic division and results in the mass of endosperm cell. So, what does endosperm do? So, endosperm ಏನು ಮಾಡುತ್ತೆ? The endosperm, it provides a nourishment to the developing embryo in the seed plant, and it also gives protection for the embryo from the mechanical injury. ಹಾಗಾದರೆ when endosperm is produced and what is its nature? See, the endosperms is produced after fertilization and it is a triploid in condition, especially in the angiosperm. So in the angiosperm, the endosperm produced is only after the fertilization and it's always triploid in nature. But in the gymnosperm, the endosperm is formed before fertilization and is haploid in nature. So gymnosperm ಏನಾಗುತ್ತೆ, endosperm is produced only before the fertilization and it is haploid in nature. Okay, so on the basis of development of endosperm, it can be classified into three types here. They are referred as nuclear endosperm, cellular endosperm, and allobial endosperm. So based upon the development, we classify the endosperm into three categories or three types here: nuclear endosperm, cellular endosperm, and allobial endosperm. Now let us study the development of free nuclear endosperm. After fertilization, the primary endosperm nucleus in the embryo sac undergoes repeated mitotic free nuclear division without any cell wall formation. So fertilization ಆದ್ಮೇಲೆ the primary endosperm nucleus, they just divide. That means the nucleus itself divide without any cell wall formation. Thus the nuclei produced appears freely in the embryo sac. ಏನಾಗುತ್ತೆ ಇಲ್ಲಿ, the nucleus just divide, so the number of nucleus is going to increase. As a result, the embryo sac will have a huge number of the nuclei. This free nuclei in the embryo sac has a liquid consistency and this can be called as the liquid endosperm. So in the embryo sac we find an enormous number of the nuclei. This free nuclei in the embryo sac, they have a liquid consistency and this can be called as a liquid endosperm. The best example is the coconut water, which is a very best example for the free nuclear endosperm. Now we will again have a glance of the development of the free nuclear endosperm. So after fertilization, the primary endosperm nucleus in the embryo sac, they undergo a repeated a mitotic division, and when they divide, they just divide only the nucleus. So once the nucleus divide, later there is no cell wall formation. Thus the nuclei produced, they appear very freely in the embryo sac. This free nuclei in the embryo sac has a liquid consistency and this can be called as the liquid endosperm. So the best example is the coconut water, which is an example for the free nuclei endosperm. So now Let us study the second type of the endosperm development called the cellular endosperm. So, the development of the cellular endosperm. So, here the primary endosperm nucleus undergoes the free nuclear division followed by cell wall formation, resulting in a mass of endosperm cells, and this represents the solid endosperm or cellular endosperm. The best example is the white part of the coconut, which we call them as a kernel, kobri. Okay, so development of the cellular endosperm. ಹೆಂಗಾಗುತ್ತೆ ಅದು? So the primary endosperm ಏನಾಗುತ್ತೆ? The nucleus of the primary endosperm, they divide. After division, there is cell wall formation around the nuclei. So this results in a mass of endosperm cells, and this represents the solid endosperm or cellular endosperm. The example is the kernel, the white part of the coconut. So now let us study the third type of the endosperm development, that is allobial endosperm. If you look the allobial endosperm development here, it is an intermediate type of the endosperm found between either, that means it is neither a free nuclear endosperm nor the cellular endosperm. So it is a intermediate type of the endosperm development. It exhibit both the free nuclear and cellular endosperm development. So in this development, the primary endosperm nuclei found towards the micropyle region undergoes free nuclear division followed by cell wall formation, resulting in a mass of endosperm. In the chalazal region, the primary endosperm nuclei undergoes only the free nuclear division. So ಏನಾಗುತ್ತೆ, ಡೆವಲಪ್ಮೆಂಟ್ ಆಗಬೇಕಾದ್ರೆ ದ ಪ್ರೈಮರಿ ಎಂಡೋಸ್ಪರ್ಮ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಇಲ್ಲಿ ಇನ್ ದ ಮೈಕ್ರೋಪೈಲ್ ಪಾರ್ಟಲ್ಲಿ ಏನಾಗುತ್ತೆ, ದೇ ಅಂಡರ್ಗೋ ಫ್ರೀ ನ್ಯೂಕ್ಲಿಯರ್ ಡಿವಿಷನ್ ಫಾಲೋಡ್ ಬೈ ಸೆಲ್ ವಾಲ್ ಫಾರ್ಮೇಷನ್ ರಿಸಲ್ಟಿಂಗ್ ಇನ್ ಎ ಮಾಸ್ ಆಫ್ ಎಂಡೋಸ್ಪರ್ಮ್ ಸೆಲ್ಸ್. But in the chalazal region, the primary endosperm nuclei, they undergo division. ಯಾವ division ಅದು? They just divide the nucleus. As the endosperm cell in the micropyle region divide further, the chalazal ends and the chalazal region gets crushed off. So ಏನಾಗುತ್ತೆ, ಎಂಡೋಸ್ಪರ್ಮ್ ಸೆಲ್ ಮೈಕ್ರೋಪೈಲರ್ ರೀಜನ್ ಅಲ್ಲಿ ಡಿವೈಡ್ ಆದಾಗ, ಗ್ರೋ ಆದಾಗ ಏನಾಗುತ್ತೆ, ಚಲಾಜಲ್ ಪಾರ್ಟ್ ಏನಾಗುತ್ತೆ, ಫುಲ್ ಕ್ರಶ್ ಆಗುತ್ತೆ ಇದು. So this allobial type of the endosperm development, it is more common in the monocot. Okay, I will repeat once again. So if you come across the development of the allobial endosperm, okay, it is an intermediate type of the endosperm, that is it exhibit both the free nuclear endosperm development and cellular endosperm development. So in this development, the primary endosperm nuclei found towards the micropyle region undergoes free nuclear division followed by cell wall formation, resulting in a mass of endosperm. In chalazal region, the primary endosperm nucleus, they undergo only free nuclear division without any cell wall formation. As the endosperm cells in the micropyle region, they divide and grow, the chalazal region gets crushed off. So this type of development of the endosperm is more common in the monocot plants. Now let us study the next concept, that is the development of a typical embryo in the plants, in the flowering plants. See, you have heard about the term called embryogenesis. So what is the embryogenesis? The process of formation of the embryo is called embryogenesis. Okay, the process of formation of the embryo is called embryogenesis. What is the importance of the embryogenesis? The embryogenesis, it usually establishes the body plan of the plant. In the early stages of the embryo, in the embryogeny, there are similarities are found in both dicot and monocot. Okay, so embryo formation ಆಗಬೇಕಾದ್ರೆ, if you compare in both dicot and monocot, they have same type of the development. So now let us study the typical embryo development in the flowering plants. The formation of the embryo begins with a division of the zygote within the embryo sac. The first division is always asymmetrical and transverse. It results in two types of the cell: suspensor cell that are found in the micropyle region and apical cell found in the chalazal region. This two-celled state establishes the polarity of the embryo. What do they establish? They establish the polarity of the embryo. The apical cell undergoes a series of division and they give rise to a proembryo. During the development of the embryo, it divides in various plane and results in different shapes. That means the embryo assumes to have a different shapes, like they passes through a stage where they have a globular shape, heart shape, and finally they mature as an embryo. They mature as a embryo having one cotyledon or two cotyledon. ಸೊ ಡೆವಲಪ್ಮೆಂಟ್ ಆಗಬೇಕಾದ್ರೆ ಏನಾಗುತ್ತೆ, ಇದು ಡೆವಲಪ್ಮೆಂಟ್ ಆಗಬೇಕಾದ್ರೆ ಒಂದು ಎಂಬ್ರಿಯೋ ಏನಾಗುತ್ತೆ, ಇಟ್ ಡಿವೈಡ್ಸ್ ಇನ್ ವೇರಿಯಸ್ ಪ್ಲೈನ್. ಡಿವೈಡ್ ಆದಾಗ ಏನಾಗುತ್ತೆ, ಎಂಬ್ರಿಯೋ ದೇ ಅಸ್ಯೂಮ್ಸ್ ಟು ಬಿ ಆಫ್ ಡಿಫ್ರೆಂಟ್ ಶೇಪ್ಸ್. ಫಸ್ಟ್ ದೇ ಅಸ್ಯೂಮ್ಸ್ ಟು ಬಿ ಎ ಗ್ಲೋಬ್ಯುಲರ್ ಶೇಪ್. Next they assumes as to be a heart shape, and finally they become a mature embryo having two cotyledons or single cotyledons. So what is the function of the suspensor cell, and what is its nature? The suspensor cell is always attached to the embryo. As a suspensor cell divide, it elongates and pushes the developing embryo deeper into the nutritive tissues. So ಏನು ಮಾಡ್ತೈತೆ ಸಸ್ಪೆನ್ಸರ್ ಸೆಲ್ ಇದು, it is always attached to the embryo. So as a suspensor cell divide ಆದಾಗ it elongates and pushes the developing embryo deep into the nutritive tissue. Okay, so now I will again repeat the development of the typical embryo. So what is the embryogenesis? The process of development of the formation of the embryo is called embryogenesis. The embryogenesis it establishes the body plan of the plant. So in the early stage of the embryogeny, the dicot embryo development and monocot embryo development are almost same. Okay, so when we come across the development of a typical embryo, see the formation of the embryo, it always begins with the division of the zygote in the embryo sac. The first division is asymmetrical and transverse, resulting in two types of the cell: suspensor cell and apical cell. Suspensor cell are found towards the micropyle region and apical cell are found towards the chalazal region. This two cell stage establishes the polarity of the embryo. The apical cell undergoes a series of division giving rise to a proembryo. During the development of the proembryo, it divides in various plane, resulting the embryo to pass through the globular shape, heart shape, and finally it becomes a mature embryo having two cotyledons or one cotyledons. So what is the function what is the function of a suspensor cell? What do they do? The suspensor cell is always attached to the embryo. As the suspensor cell divide, it elongates and pushes the developing embryo deep into the nutritive tissue. So now let us study the structure of the dicot embryo. If you look the dicot embryo, it consists of an embryonal axis and two cotyledons. The portion of the embryonal axis found above the cotyledon is called epicotyle. So what is the function of the epicotyle? Epicotyle ಏನು ಮಾಡುತ್ತೆ? It develops into the plumule or the stem part of the plant, and the part found below the cotyledon is called hypocotyle. This hypocotyle, it always develops into radicle or the root tip of the plant body. Okay, if you come across the structure of dicot embryo, the dicot embryo have an embryonal axis and two cotyledons. The portion of the embryonal axis above the cotyledon is called epicotyle that develops into the plumule or stem part of the plant body, and the portion found between the cotyledon is called hypocotyle that develops into the radicle or the root tip of the plant body. So now let us study the structure of the monocot embryo. If you look the monocot embryo, it has only one cotyledon called the scutellum. So monocot embryo they have only one cotyledon and it can be called as the scutellum, especially in the grass. And the embryonal axis found above the scutellum is called epicotyle that develops into the shoot apex and leaf primordia. The shoot apex and the leaf primordia are completely enclosed in a hollow foliar structure called coleoptile. So ಏನಾಗುತ್ತೆ ಈ ಎಪಿಕೋಟೈಲ್ ಇರುತ್ತಲ್ಲ, this epicotyle, it develops into a shoot apex and the leaf primordia. Both the shoot apex and the leaf primordia are enclosed by a hollow foliar structure called coleoptile, and the portion of the embryonal axis found below the scutellum is called hypocotyle. This hypocotyle part develops into the radical part of the plant body. That means it develops into the root cap of the plant body. So the radicle and the root cap are completely enclosed in a sheath called coleorhiza. I will repeat once again. The monocot embryo has only one cotyledon and it can be called as the scutellum in the grass. The embryonal axis found above the scutellum is called epicotyle that develops into root apex and the leaf primordia. The shoot apex and the leaf primordia. The shoot apex and leaf primordia are completely enclosed in a hollow foliar structure called the coleoptile. The portion of the embryonal axis found below the scutellum is called hypocotyle that develops into the radicle. The radicle gives rise to the root cap of the plant body. So the radicle and the root cap are completely enclosed in a sheath called coleorhiza. So now let us study the next concept about the seed. So we will look what is a seed and what is it made up of. Seed is a matured fertilized ovule. It contains an embryo, the reserve food material, the cotyledons, and the seed coat made up of tegum and tegum. The cotyledons are embryonic leaf of the seed, which develops into the first leaf of the embryonic plant. The reserve food are stored in the endosperm that are used in the embryo during its germination. So what is a seed? Seed is a matured fertilized ovule that contains embryo, the reserve food material, the cotyledons, and the seed coat made up of testa and tegmen. The cotyledons are embryonic leaf of the seed, which develops into the first leaf of the embryonic plant. The reserve food materials are stored in the endosperm that are used by the embryo during the germination of the seed. Now, let us study the types of the seeds based upon the presence or absence of the endosperm. We classify into three categories here. They are named as albuminous seed, non-albuminous seed, and the perisperm seed. Okay, number 1, albuminous seed. Number 2, non-albuminous seed. Number 3, perisperm seed. So what are albuminous seed? Albuminous seed are those seed which have the endosperm. The example is the castor seed, the poppy seeds, some of the cereals, the millet, and the sugar cane. So what are non-albuminous seed? The non-albuminous seed are the seed which do not have the endosperm. The best example is the mustard, these the gram, pea, halicima, and the mustard. So in the non-albuminous seed, they do not have the endosperm. In the albuminous seed, they have the endosperm. We have the third category called the perisperm seed. So what are perisperm seed? These perisperm seed do not have the endosperm, but they have a small part of the nucellus that acts as a nutritive tissue. So the perisperm seed or the seed which do not have the endosperm, but they have a small part of the nucellus that act as a nutritive tissue, and this can be called as the perisperm. The best example is the black pepper and the beet. So ಏನಾಗುತ್ತೆ, the perisperm speedily endosperm ಇರೋದಿಲ್ಲ, but they have a nutritive tissue called the nucellus. Now let us study the next concept about the fruits. So what is a fruit? The fruit that usually develops from the ovary are called the true fruits. Example, the cucumber, the tomato, the coconut. So ಯಾವುದೇ ಒಂದು ಫ್ರೂಟು ಓವರಿಂದ ಡೆವಲಪ್ ಆದರೆ they're called the true fruits. Then what is a false fruit? If the fruit that develops from other part of develops other than the ovary, especially the thalamus, they're called the false fruit. So if the fruit that develops other than the ovary part, example thalamus, it is called false fruit. Example, the apple, the strawberry, and the cashew. We have the third category of the fruit, they're called parthenocarpy fruit. What are parthenocarpy fruit? When the fruit that develops without the fertilization of the ovule is called parthenocarpic fruit, and the phenomena is called, the condition is called parthenocarpy. The parthenocarpic fruits are always seedless fruits, and they can be induced artificially by spraying a synthetic growth hormone. The best example for parthenocarpic fruit is the banana and the grapes. I will repeat once again. So we can classify the fruits into three categories here. The fruit that develops from the ovary are called the true fruits. Example, the cucumber, tomato, and coconut. And the fruit that develops other than the ovary, that means the thalamus, okay, they're called false fruit. Example, the apple, the cashew, and strawberry. And if the fruit that develops without the fertilization of the ovule, it is called parthenocarpic fruit, and the condition is called parthenocarpy. The parthenocarpic fruits are usually seedless fruits, and they can be induced by spraying some synthetic growth hormone. The example for the parthenocarpic fruits are the banana and the grapes. So if you take the entire flower, so which part of the flower will produce the fruit and the fruit wall and the seed? So usually the ovary wall will produce the fruit wall. The ovary is going to develop into a fruit. The ovule develops into the seed. The integuments, they develop into the seed coat. So now let us study the last concept of this chapter, the special mode of reproduction. As we know, in the reproduction there are two categories here, asexual and the sexual reproduction. But there are some kind of reproduction where they are neither sexual nor asexual, and this can be called as the special mode of reproduction. In the special mode of reproduction, we have two types here. Number one, apomixis. Number two, polyembryonic. If you look the apomixis, so what is the apomixis? Apomixis can also be called as agamospermy. So in the apomixis, there is production of the seed without the fertilization. So apomixis means the process of production of the seed without the fertilization is called apomixis, and the seed is called apomictic seeds. So in angiosperms, over 300 species from more than 35 families are said to be apomictic in nature. So in the angiosperms, over 300 species from more than 35 families are said to be apomictic in nature. There are several ways to develop the apomictic seeds. In some of the species of Asteraceae and Solanaceae, the diploid egg cell directly develops into embryo. ಇದು ಒಂಥರ ಆಗುತ್ತೆ. ಮತ್ತೊಂದು, in some of the species of onion and lily, the synergids, antipodal cells and the nucleus cells, they give rise to embryo. I will repeat once again. So apomixis means it is the process of production of the seed without the act of fertilization is called apomixis, and therefore the seed produced through apomixis are called apomictic seed. In angiosperms, over 300 species that belongs to 35 families are said to be apomictic in nature. There are several ways of development of apomictic seeds. In some species of Asteraceae and Solanaceae, the diploid egg directly develops into the embryo. So in some of the species of Asteraceae and Solanaceae, the diploid egg directly develops into embryo. But in some of the species of onion and lily, the synergids and antipodal cells and the nucleus, they give rise to the embryo. Now we'll discuss the next type of the special mode of reproduction, that is polyembryony. So what is polyembryony? Polyembryony means the development of more than one embryo in the seed is called polyembryony. So the development of more than one embryo in the seed is called polyembryony. The polyembryony was discovered by Anton von Leeuwenhoek in the citrus plant. It is more common in the gymnosperm, especially the conifers, than the dicot and the monocot. So gymnosperms are tumba common ಆಗಿರುತ್ತೆ. See, there are two types of the polyembryony. True polyembryony. The other is the false polyembryony. What is the true polyembryony? If the multiple embryos they develop from the egg, it is from the egg in the embryo sac, it is called true polyembryony. So true polyembryony means if multiple embryos develops directly from the egg cells in the embryo sac, it is called true polyembryony. There are two conditions here in the true polyembryony. If the zygote divide and splits into multiple units, each unit will develop into embryo, and this condition can be called as the cleavage polyembryony. So zygote ಏನಾಗುತ್ತೆ, divide into number of small unit. So each unit that develops into the embryo, and this is called cleavage polyembryony. Simple polyembryony andre ಏನು, when there is more than one embryo sac in the ovule, it results in the development of multiple embryos. This is what we call simple polyembryony. So when there is more than one embryo sac in the ovule, it results in the development of multiple embryos. This is what we call simple polyembryony. Now let's study about the false polyembryony. What is false polyembryony? If the multiple embryos develops from other than the egg cell, that means the nucleus or synergids or integument and antipodal cells, it is called false polyembryony. So if multiple embryos they develop from the synergids, antipodal cells, nucleus and integument, it is called false polyembryony. When the nucleus or the integument cells are involved in development of embryo, they are called adventive polyembryony. So nucleus or integument cells, if they are involved in development of the embryo, it is called adventive polyembryony. If the synergids and antipodal cells are involved in producing the multiple embryos, they are called mixed polyembryony. Okay, so in the false polyembryony, we have two conditions here: advent polyembryony and mixed polyembryony. Advent polyembryony ಅಂದ್ರೆ ಏನು? If the embryo develops from the nucleus and the integuments, they're called advent polyembryony. So what is a mixed polyembryony? If the embryo develops from the synergids and the antipodal cells, they're called mixed polyembryony. So I will revise, I will repeat once again. So polyembryony means that the development of more than one embryo in the seed is called polyembryony. It was discovered by Anton von Leeuwenhoek in the citrus plant. It is more common in the gymnosperms than in the dicot and the monocot. There are two types of the polyembryony: true polyembryony and false polyembryony. If the multiple embryos develops from the egg cell, it is called true polyembryony. If the multiple embryos develops other than the egg cell, for example synergids, antipodal cells, or the nucleus, it's called false polyembryony. In the true polyembryony, there are two conditions here. Number one, cleavage polyembryony. Number two, simple polyembryony. So what is a cleavage polyembryony? If the zygote splits into multiple units and each unit develops into embryo, it is called cleavage polyembryony. Simple polyembryony means what? If there is more than one embryo sac in the ovule, it results in the development of multiple embryos. This is called simple polyembryony. In the false polyembryony, again there are two conditions here: advent polyembryony and mixed polyembryony. If multiple embryos develops from the nucleus and the integument, it is called advent polyembryony. If the multiple embryos develops from the synergids and the antipodal cells, they are called a mixed polyembryony. Okay, so now let us study the importance of apomixis and polyembryony. If you look in terms of the significance of both apomixis and polyembryony, they're almost the same. If you look the apomixis, okay, so apomixis has a very great genetic value because it produces a homozygous plant that retains the characters of the plant for several generations. If you look the polyembryony also produces genetically similar homozygous plant, and it has a very great application in the horticulture and agriculture. Okay, so with this we have finished the second chapter, sexual reproduction in the flowering plants. Okay, so thank you.